everyone, how are y'all? I hope you all are doing amazingly well today, because today we're going to not be studying but actually clearing some things, explaining something, and having so much fun. So I think that today's video is going to be smaller than the previous one. And if you don't know, that this is my camera checkpoint, 3rd edition lower secondary science, grade 8 book, and the author of it is Peter D. Riley, okay, of this really thick book. So yeah, okay guys, so it's going to be really interesting. You know that it's divided into 4 sections if you watched my last video. So we're going to be continuing my second chapter from like the 13th page. Actually it was 15 because we already read 2 pages and a lot of concepts. So yeah. So after the willow tree experiment and its questions, we have the scientific revolution, and then it's shown that the work of Stephen Hales is an example which built on the work of this person. I forgot what his full name was. It was a Belgian scientist, and his full name was apparently Johannes Baptista van Helmont, but we can just call him Van Helmont. So Stephen Hales and I guess Jan Ingenhousz further evaluated his idea. Now I know that a lot of people would think that this is copying. I thought it was copying at first, but to be honest, they were just trying to improve. And although it was his plan, but he did something, and now he also made the theory of how do I say this? He made the theory of air, impacting plants. And his time was 100 years after the scientific revolution and after, Van Helmont. So this English scientist, he was Belgian, this is an English scientist. At his time, it was known not just 4 elements, but actually the breakdown of air and water, and that water is a compound and air is a mixture, and also about, carbohydrates, like carbon, hydrogen, oxygen, all other stuff, meaning actual elements. So all this information led to a review of Van Helmont's idea, and then he only proposed a theory that, air impacted plants, but this person, Jan Ingenhousz, was the one who actually, found out stuff. He actually was the one who found out that green plants were the ones that could only do it, like chloroplasts, the organelles, and it only happened in leaves. And he also found out that it only happened during the daytime, because we all know that photosynthesis only happens in the day. Why does it happen in the day? Because plants need sunlight to make food, and in the night, respiration happens. So yeah. In the night, just respiration can happen because photosynthesis cannot happen. So he found that out, and he found that green plants were the responsible ones, and he also experimented and actually was the one who, said, okay, this is true, that air is also needed. He was the one who, how do I say this? Proved this scientist right. And obviously water was already proved right through Van Helmont, but yeah, he wasn't the one who found that out. So these are all the questions. I answered this one. I answered this one too. Why did I review of Van Helmont's work? We all know this. It was because air was the only other thing that was impacting plants other than water, especially at that time, as much as people knew. So why did our review of Van Helmont's... Okay, how did Ingenhousz's discovery suggest that carbon dioxide might be important in making food? Obviously, it's kind of a similar answer to this one. And which 2 activities in step 3 of making a scientific inquiry were being used in reviewing Van Helmont's work? I'm sorry for saying that so quickly. But this happened in these pages, and I would say it was observation and interpretation. If you're wondering what interpretation is, it's curiosity, and observation is just observing and looking at stuff. So yeah. So after studying a little history of the science, it's now your turn to do experiments. And by your turn, obviously, I mean our turn. But I don't have a laboratory right now, so I guess you guys are going to do it first. So you can also experiment this idea in your laboratory, And one more, that light is needed for plants. You know that if you keep a plant in darkness, then it can't survive, but it can survive for a small amount of time. And how does it do that? It does that using starch inside it. Now you may ask, what is starch? You didn't tell that to us before. Well, starch is just another type of sugar like carbohydrates and glucose. So yeah, it's one of the other types, and it's like carbohydrate, it's not the smallest. Glucose is the only one which is the simplest sugar. So the plants use that when they're in darkness to survive for a little amount of time, because without light, plants normally die. And if you actually keep them for a long period of time, their starch is finished and they die. But if you do that for only a few days, then the starch is still there and you can test if the plant has starch or not. And how do we test it? Perfect question. We're going to come to that now. But first, look at these maize plants in southwest France that died through lack of water in a drought. Look at them, so dried up. Do you feel sorry for them? Because I do. And this is kind of a summary of chapter 1, which I can do for you. Because like water does travel through a plant and is scraped through the leaves through a transpiration stream. So what happens is that there's a plant and the root of the plant, like here's the root, it absorbs all of the water that comes, and then it passes through the stem, and then it goes into the leaves, and then there it some is used and some is transpirated, like some evaporates. So that is called transpiration. Now obviously when it evaporates, then more comes, and it just happens and happens and happens again and again. So that is called the transpiration stream. But if you don't understand, no worries, because you can watch my chapter 1 video. Okay, so testing for the carbohydrates called starch and leaves. Finally, now this is a diagram showing starch grains that form in the cytoplasm of leaf cells, which also contain chloroplast. So these are starch grains. So figure 2.9 shows the distribution of chloroplast in the cells of leaves. So these are the cells of a leaf, and these tiny dots, green dots, are chloroplasts, and this is the nucleus, this gray one. Now let's finally test for starch, guys. Okay, so the first thing is, you don't have to read all of this, just work safely, okay? So first, all you have to do is put like the leaf into a beaker of water for 1 minute. That way it gets soft and it kind of dies. Now don't worry, it's not just cruelty, but it's actually so that, all of the enzymes die and the iodine solution can be seen properly. So then you use a pair of forceps, because obviously it's hot, and then you put it in a test tube like this. A test tube filled with ethanol, so that you can kill the chlorophyll, aka the green pigment in the leaf. And the reason why you just don't heat up the ethanol, because ethanol is alcohol and it's inflammable, so you don't want to cause a fire. So remember, first put it in a beaker of hot water and then put the test tube inside, and then only heat. Now what does that do? It kills the chlorophyll, but it also makes the leaf very brittle, which means it's crispy and hard, if you were wondering what brittle means. So it's kind of, a risk of breaking the leaf when you're putting iodine on it. So you boil it again, and when you boil it again, it becomes soft again. So after that, what you can do is finally put the iodine solution on top of it. Okay, iodine solution is a solution in which you can use to see if starch is on a leaf. So first put it on a smooth surface, a white tile or any smooth surface you have, and then drop the iodine solution, a few drops, and if they are stained blue, black, then there's starch present. And if it isn't present, then it will stay the same color. Or you can say green yellow, because that's normally the colors of leaves who don't have chlorophyll anymore. So yeah, it's kind of sad that the chlorophyll is gone and you did all of that, but that the plant was kept in dark, or if it wasn't kept in dark. So yeah, in the dark for enough time to finish all of the starch. So here are 2 questions related to it. Plant cells build up starch and then remove it, as you shall see later. When testing a leaf for starch, what might happen if the cells were not killed? I already told you that the enzymes will not die, and then the color will not show properly of the iodine solution. So obviously it will disrupt the process and the experiment, although it's kind of cruel. What would happen if the boiling point of ethanol was higher than that of water? I just told you, it would be inflammable. And obviously, it'd be the same thing kind of as just, putting the ethanol, heating the ethanol, because, once again, it will cause a fire. Now, these are 3 more questions. What color would you expect the ethanol to go after the leaf has been boiled in it? Green, yellow. I already told you. So it's kind of a creative question. Like, what color would the ethanol go? So I don't really know the real color of ethanol, so you can search this one up. What might happen in the brittle leaf if you try to spread it out on the white towel? Yeah, it would break. That's what I told you. Why is it important to spread the leaf out on a white towel and not a colored one? So that you can see the color properly. That's pretty obvious. Okay, guys, so I guess that's enough studies for today. I hope you had a lot of fun, because if I'm being serious, I had a little more fun today because I found everything way more clearer than before. So today, you learned a lot of stuff. You learned the starch experiment, you cleared all your queries towards it, you just saw what would happen without water, you learned about 2 more new scientists and what they did, and like the difference between them and Van Helmont. And not just that, but you learned what starch is as well. And once again, you reviewed what photosynthesis is, like what it needs. And trust me, that's really important to keep reviewing what you study, because the human brain normally forgets. So guys, that's it for today. We're going to start back on page 19. That will be part 3. So stay tuned. Don't forget to like, share, and subscribe if you liked my video. And bye!